All right, welcome to Life and Biology. I'm Dr. Joel Graff, and I'm going to go over the slides that were presented in my lecture, Principles of Living System lecture on January 24th, 2022. We have learned a little bit about chemistry, a little bit about organic chemistry, a little bit about water chemistry, and now we're moving on to biochemistry. And in biochemistry, we've got four main biological macromolecules to start off with and start to wrap our head around. And these molecules, with the exception of lipids, will form polymers. So that would be proteins form polymers, nucleic acids are polymers, and so are, carbohydrates. They can be polymers, polysaccharides. And so we have some lingo here where we have monomers, dimers, trimers. So 1 part, 2 parts, 3 parts. And so for the next slide or two, I might be using this kind of a notation. We've got some part, whether it's a nucleotide or an amino acid or whatnot. And then you've got another nucleotide or amino acid or carbohydrate, and they're connected together with a covalent bond. So in a disaccharide, you'd have 2 monosaccharides connected together with a covalent bond. So we want to talk about how you can go from having a bunch of monomers, single parts, to having a polymer. So here's just an example. If we had a molecule that was currently a trimer and we wanted to add a monomer to it, we would take those 2 molecules, add them together, and in a reaction that is reversible, we would move our reaction in the right direction following this arrow, and then we would end up with a tetramer. So you can see there's now 4 parts in the molecule where there used to be just a 3-part and a 1-part separate. This reaction is reversible, and there's a key component of water that water plays in this reaction. So if we go from small parts and build them together into a larger part, that's called a condensation reaction. It's also known as dehydration. Sorry, this handwriting looks terrible, but it's a little bit better when it's not converted from the smartboard. And when you do a condensation or dehydration reaction, what you're doing is you're removing a hydroxyl group and a hydrogen proton, from the beginning 2 things in your reaction, which are called substrates. You pull the water out of it, and then there gets a covalent bond formed between the monomer and the trimer, for example. So just like you can pull a water out to cause covalent bonds to form between your parts of your polymer, if the reaction goes in the reverse direction, you can go from the 4-part and then a water would go into the reaction, and then you could break one of those covalent bonds, and you could be back to a trimer and a monomer, or a dimer and a dimer. So it just depends on where the parts of the water, the proton and a hydroxide ion, where those would get inserted, is how you would is where the break would occur within these covalent bonds holding your tetramer together. So we're making bonds going in the right direction, rearranging bonds actually, pulling a water out and connecting the pieces together. If you go from right to left in this reversible reaction, you're going to be breaking a bond within your growing polymer molecule, multi-part molecule, and you can break them apart. And because water is going in, we call it hydrolysis. So condensation reactions are when we go from small parts to larger parts, and hydrolysis reactions are when we go from a large part to smaller parts. Now we have because it's breaking using water, that's, if you look at the word hydrolysis, hydro is for water, lysis is the action of breaking, and so this is using water to break apart your tetramer. Okay, so then on the next slide, we will talk about the biological macromolecules, carbohydrates, or carbs for short, lipids, nucleic acids, and proteins. We're going to walk through each of these in lecture, and we're going to start off with the carbohydrates. But just to give you an overview of where we're going with all this, we want to look at each of these 4 categories of biological macromolecules and think about think about what we would call the monomers, dimers, oligomers, which means few parts, and polymers, many parts. So, dealing with carbohydrates first, we start with monosaccharides. That's what the monomer is called. A dimer is called a disaccharide. An oligomer of sugars is called an oligosaccharide. And then once you get to polymers, we call it carbohydrates. We could also call it a polysaccharide. So if you ever hear polysaccharide, that's valid too. So those they work pretty well with the prefix and then just throw saccharide at the end. When we get to lipids, this doesn't really make sense to call it a polymer because there aren't covalent bonds formed between each of the individual lipid parts, the lipid molecules, and they do cluster together within a water environment because lipids tend to be, for the most part, nonpolar, and the nonpolar parts of the lipids will want to hide or cluster to reduce their interaction with water because they are hydrophobic. Okay, so there are 4 categories, and we'll walk through those in the next lecture, but there's lots of variation within these categories of the 4 types of lipids. Moving on, nucleic acids. The polymer is called a nucleic acid, just like the copolymer of proteins are called proteins. The polymers of carbohydrates are carbohydrates, and polysaccharide is actually the more technical correct term, which I didn't put on the slide, so sorry about that. But the parts that make up nucleic acid are called nucleotides. And for DNA, if we want to build DNA, we have 4 different parts, which are A, T, C, and G. If we want to build RNA, we have 4 different parts, A, C, G, and U. And we'll get to that later. If you have 2 nucleotides stuck together, it's called a dinucleotide. Oftentimes you can type in some DNA sequence into your computer and send that information off, and there will be a machine that will make oligonucleotides for us. We do this in the lab all the time. Your cells also make oligonucleotides. Just whenever you have, say, 5 to 100 nucleotides, we'll call it an oligonucleotide. And then at some point, the magic number of nucleotides gets so long that we call it a nucleic acid. Okay, so those are your parts of monomers, dimers, oligomers, and polymers for nucleic acids. Now for proteins, the final biological macromolecule that we'll dig through in this lab. And these are the building blocks for proteins, or the monomers for proteins, are called amino acids. And so there's 20 different amino acids that we will discuss. Dimer of amino acids, you can have dipeptides. And then oligomer is a peptide. And then when you have a polymer, it's called a protein. So we have different ways, different categories to describe how big of a molecule built out of amino acids we have. So again, just like oligonucleotides, there's not a real cut and dry definition of what when a peptide gets to a length that you would then call it a protein. So it's a little subjective on where that cutoff is. Okay, carbohydrates. Carbo indicates that there's going to be carbon in these molecules, and hydrates indicates that there might be some water. And so if we jam that all together, we get CH2O. And carbohydrates have different numbers of carbons, and so depending on the number of carbons, we'll multiply the H2 and the O by that number of carbons. So for example, if you have a 3-carbon sugar, we're going to call it a tri for 3, os for sugar. A typical triose might have a formula of C3H6O3. And what we did to get to this, again, is just you take the number 3, you multiply it by your carbons, the 3 times the 2 hydrogens, and the 3 times the oxygen, and you get C3H6O3. So then, just like if we were going to multiply 3 by this carbohydrate core, pentose sugars also will follow that, but this time pent means 5, so we have 5 carbons. H2 times 5 is H10, and O times 5 is O5. So pentose oftentimes will have C5, H10, or O5, somewhere in that ballpark. Later we're going to talk about glycerol, and that's a trio sugar, and it is actually C3H8O3. So they can vary from this this sort of just take the number times CH2O and you get the answer. However, we're going to mostly focus on hexoses right now. We'll come back to pentoses when we talk about the ribose that is within nucleotides. But for now, we're just going to focus on hexoses. And so hex means 6, os means sugar. So a 6-carbon carbohydrate. So here we've got C6, H2 times 6 is H12, and O6. So the example we'll come to again and again for a hexose is glucose. Now the product of the glucose is the product of photosynthesis. We can take some carbon dioxide out of the air. Well, we can't. Plants can. Any photosynthetic bacteria or algae or anything like that can do this. They take some carbon dioxide out of the air. They take some water, combine it together, and out pops glucose. And so it's also, when we go to move on to talking about cellular respiration later on in the course, we're going to be taking a glucose and then breaking it back down. So whether we're building up glucose in photosynthesis or breaking it down during cellular respiration, we're going to be dealing with glucose a lot in this class. So that'll be our one that we spend the most time on. However, there are some hexose monomers I'd like you to know, and that includes 3 that we will discuss in biology, this POLS class. One is glucose, which we've already mentioned, fructose, and galactose. Now, even though these are different sugars, they all have the formula C6H12O6, and that's because these are just, if you rearrange the carbon and hydrogen and oxygen molecules, you can convert from one sugar to the other. In fact, converting from a glucose type molecule to a fructose type molecule happens as soon as glucose enters a cell, and so, or it's the second thing that happens technically. And so it's just a rearrangement. So those are isomers of each other. Okay, so those were the 3 monomers that we were going to talk about. I'd like you to know 3 dimers as well. And so a dimer of sugars is called a disaccharide. And here we've got sucrose, maltose, and lactose. If you break down sucrose with the enzyme called sucrase, so SUCR lets you know you're dealing with sucrose, SUCR, and then ASE lets you know you're dealing with an enzyme. So oftentimes enzymes end in the suffix ASE. And what these enzymes do is they speed up the reaction. So if you want to break down sucrose faster, sucrase is a catalyst that'll help you do that. It's a protein. There's a lot of proteins that are catalysts, and again, they're called enzymes. But if you break down a disaccharide of sucrose and look at its building parts, or its 2 monomers that were put together to make the sucrose molecule, you get 1 glucose and 1 fructose. Moving on to maltose, the enzyme is maltase, so we're following that same thing. Malt, os for sugar, malt, ase for enzyme. And so then you end up with 2 glucose molecules when you break that down. And then finally, lactose, and you need lactase to break that down. If you're lactose intolerant, your body stops making this lactase enzyme, and you need to take a pill with your dinner, any meal you have that has dairy in it, because lactose is very common in dairy. And you end up with a glucose and a galactose. So for these 3 dimers that we want to know, they all have 1 subunit that is a glucose, and then the other subunit is the fructose, glucose, or galactose, which were the 3 monomers I wanted you to know. So we're not going to go real heavy into carbohydrate biology and the different types of sugar molecules you can have, but this is the amount I'd like you to know for quizzes and that sort of thing. Also, a good quiz question is, what's the enzyme that breaks down maltose? And hopefully you can just remember that you just need to take the ose, change it to ACE, and you've got the enzyme. Okay, so let's focus a little bit more on glucose again. This slide is a little bit busy, but when I drew it in class, I focused on the fact that we had a 6-carbon sugar. So we can go, here's one carbon, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. When glucose is dry, it tends to form a linear arrangement like this. And what happens when it's in the linear formation, we've got a carbonyl group at the end. Just a side note, when a carbonyl is at the end, it's called a ketone. In fructose, the carbonyl is actually on the 2-carbon, and because the carbonyl is not on an end group, fructose is what's known as an aldehyde because it has a carbonyl not on an end carbon. But anyway, going back to what happens as we take this molecule and we put it into water. Remember, we have to think about what happens with all these molecules we talk about when they're in the context of water, because water is found everywhere with life. And what happens is, because this oxygen here on the 5-carbon is electronegative, it's going to be hogging some of the electrons from that proton atom that's linked on there. And because it's going to have a partial negative charge, this molecule can bend around, and the negative charge that's built up on the oxygen on the 5-carbon hydroxyl will then do an attack, is what it's called, on the carbon here. Why did it attack this carbon? Well, it's because this carbon has a partial positive charge to it, and that's because this oxygen is being a hog of electrons. It's being greedy and keeping the electrons most of the time. So we have a partial positive charge here. So when you take this partial negative charge, attack this carbon, you get a covalent bond formed between those two. Where did the electrons come from to get that bond? Well, it came from this hydrogen, and a hydrogen without its electrons is a proton, which we designate H+. And then up here, when you form a bond with this carbon, now that carbon had 5 shared bonds around it. That's too many. The oxygen, being electronegative, will take up the slack, and if there's a proton floating around, it'll form a bond with a proton. And so, although we gave up a hydrogen down here, the proton was released down here. Up here, where the electron, we had extra electrons in the end, they formed a bond to a hydrogen. So let's take a look over at the circular form of glucose and see what it looks like once that circularization reaction happens. So here's the 1-carbon again. Here's our 1-carbon. This carbonyl group is no longer carbonyl. It's going to be a hydroxyl group, right? Because it's going to be a single bond between the carbon and the oxygen, and a single bond between the oxygen and the proton that it formed. And so that is designated right here where we've got the hydroxyl group. And note for a second that it's pointing down. I'll come back to that later, but it's not always pointing down. But let's go to the 2-carbon. On the 2-carbon, there was a hydroxyl and a hydrogen. That's what we have here. The 3-carbon, we have a hydroxyl and a hydrogen, and we've got that here. 4-carbon, we've got a hydroxyl, or a hydrogen and a hydroxyl. And so then we've got that shown here. And on here, it's always pointing in this direction down. Number 5, we had this carbon that was connected to this oxygen is now connected to the 1-carbon. So here's the 5-carbon. There's that connected to that oxygen, and this is the one that did the nucleophilic attack. It made the bond with the 1-carbon, and so now it's circularized the sugar. Okay, and then finally we've got the 6-carbon, and so it's got a hydroxyl group and 2 hydrogens. And so if we look at this one, we've got a carbon with a hydroxyl group and 2 hydrogens. So we've accounted for all of the atoms that were here, and they are over here as well. Coming back to this point where I had the hydroxyl group pointing down, when it points down, it's called alpha glucose, but it just depends on how this circularization works, how the position of the hydroxyl or the carbonyl group at that time and the hydrogen group at that time, how they were positioned. In half the time, it's going to have the hydroxyl pointing down, and the other half the time, when this attack is made, the hydroxyl group will be pointing up. So just keep that in mind, because next we're going to talk about what is the significance of having the carbon being up or down. And so here's an example where you have 2 different alpha glucoses. That means the hydroxyl is pointing down. I summarized the molecule by not drawing out every carbon and oxygen. So we know that whenever there's a point and a change in direction, it's a carbon there. And we know if the carbons that we can see don't have 4 bonds, like this first carbon, we only see 1, 2, 3. We understand that there's a hydrogen there that we just were too lazy to draw. Okay, so anyway, the hydroxyl was pointing down, so it's alpha glucose. And then we've got another one here. And what we're going to do is we're going to try to connect the 1 carbon here to the 4 carbon here. So it's going to be a 1,4 linkage. When we form a covalent bond between 2 monomers, like we talked about in the very first slide, we can suck out, or a water gets pulled out of the reaction. So it's a dehydration or condensation reaction, whichever term you prefer to use. And so to get that water, we needed a proton and we needed a hydroxide ion with a negative charged OH. And so you can see I underlined an OH here and an H here. So oftentimes if you can find an OH, 2 OH groups, that's a chance for a bond to be formed between 2 glucoses or 2 sugar molecules. You pull out the H2O, this H, that H, make the H2, and then this O is the oxygen. And then this oxygen that was not underlined, it now has a covalent bond to the 1 carbon. So your final molecule, the disaccharide from these 2 glucoses, will look like this, where it's a 1,4 linkage like we talked about, because that's the 1 carbon and this is the 4 carbon. And then it also has an alpha to indicate that when the 1 carbon sugar was an alpha glucose when it made this bond. And so then you get a dip between those 2, connecting those together. All right. So then here is the alternative, and that is if you've got a glucose, whether it's alpha glucose or beta glucose on the right, we're more interested in the beta glucose, or the glucose on the left. This time it's beta, which means that the hydroxyl on the 1 carbon is pointing up. And then if we take a hydrogen and a hydroxide, or a proton and a hydroxide ion out of this, the oxygen here will form a covalent bond to the 1 carbon. So it's the same kind of linkage that we had last time, which means it's a 1,4 linkage. But the difference is that when the glucose had the oxygen up here, so this first sugar, this first glucose, we had to draw up to get to the oxygen, and this sugar we draw down because it's coming out of that 4 carbon and it's always pointing down. And so the linkage looks like this. And so now we've got a linkage that goes up to the oxygen and then up to the 4 carbon. And if we go back here, we had down to the oxygen and then back up. So there was a dip. And so hopefully you can see that those are different. And so that just starts to give you an idea of how you can have different sorts of polysaccharides. Even if you're only dealing with glucose, you can start arranging these in various ways. And finally, you could take any of these hydroxyls, whether it's on the 6 carbon up here or down on these other carbons, and they can link together to other glucose molecules. And so there's a lot of variation that can happen with your linkage of monosaccharides. Okay, that's all we covered on that day in the lecture. It took a long time in the lecture because we drew those out. If you're following along, you might have had to pause to draw those out. If you haven't, or if you followed along with the lecture just to make sense of the lecture notes, now is time to draw out those molecules so you can kind of get it in your brain what those look like. Anyway, again, this was Life and Biology. It was a condensed lecture from my Principles of Living Systems lab, and you're more than welcome to subscribe or not. I'll talk to you later.